Today we're going to discuss about the classification of fungi in diversity of living world, Unit 1, Chapter 1, Botany. And we have discussed already the general characteristics of fungi and the reproduction or methods of fungi. Now we're going to see the classification of fungi. So the classification of fungi is based on the vegetative and reproductive characters. So there is a traditional classification of fungi. It is classified into four classes: Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes. The Phycomycetes, the word phyco means it refers to the algae. So that means in this class it has an origin of algae. The fungi is arise from algal origin, or it going to give to the algal origin fungi in this. So some of the fungi resembles the algae. Okay. And in this you have subclasses Oomycetes, Chytridiomyces and Zygomycetes. These are the subclasses seen in Phycomycetes and they are lower fungi. Okay. So traditional classification, they are divided into four classes: Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. Next, the Alexopoulus and Mims in 1979 in their book Introductory Mycology, they classify the fungi into three divisions: Gymnomycota, Mastigomycota, Mastigomycota. These are the three major division. From this three division, they divide into 8 subdivisions and 11 classes and one form class. From that 3 form subclasses, one form class and 3 form subclasses. So keep it in mind, there are 3 main divisions. From that you have 8 subdivision, and from the 8 subdivision you have 11 classes. That means 1 forms the class and 3 forms the subclasses. Oomycetes. So Oomycetes, we're going to see the general characteristics of the Oomycetes. Oomycetes mycelium is a cenocytic. So cenocytic means there is no cross wall. You can see in this structure of 5A, there is no cross wall and the nucleus is scattered. So many nucleus are seen, no cross walls. So this type of condition is called cenocytic mycelium. And the cell wall is made up of glucan and cellulose. The cell wall of the Oomycetes is made up of glucan and cellulose. Zoospores. One is whiplash type and another one is tinsel type of flagella. So you have studied zoospore in the methods of reproduction and it is a motile one. Okay. So one will be like this. This is called whiplash. That is whiplash type and tinsel type. So one will be rough and one will be smooth. So this will be rough and this will be smooth. Like this, 2 types of flagella is seen in the zoospore. Sexual reproduction is oogamous type. Oogamous means what? Oogamy means what I said. Morphologically and physiologically different. Both the gametes are different. Antheridium and oogonium is seen. So that type of sexual reproduction is called oogamous type. Example, albigo. So this is the albigo. You can see this in spinach leaves, green leaves. At the back side of the leaf you can see this white patches, that is called albigo. When you buy the leaves, green leaves, you turn to the back side and you can see this white patches, and that white patches is called albigo. If you take a section, it will be like this. So in the leaf, in between, the hyphae will be seen in the section. Next we move on to Zygomycetes. In Zygomycetes, most of them, that is, it has saprophytes and parasites. Saprophytes means live on dead organic matter. The fungi lives on dead organic matter. And parasite means it lives in the host and creates disease to the host. Example, Entomophthora on housewives. Entomophthora is a fungi, lives in the housefly. Bread mold fungi. Example, Mucor and Rhizopus. Bread, it lives on the bread. Or you can keep, instead of, that is, in your house, they had you have prepared idli, no? That idli can be keep it outside. That is, for 2 days. The second day, you can notice a black color, that is a black color layer is seen on the idli. That is called bread mold fungi, Mucor or Rhizopus. The Rhizopus is the best example which grows on idlis and bread. And Mucor is also. So you try it with your idli. Please don't waste the entire idli. A little bit of idli, you put it in your home outside. That is, outside means in the kitchen. You keep it on a plate or anywhere else. You can see after 2 days you can see the fungi which is called a bread mold fungi. Corpophilus fungi. Corpophilus means this fungi grows on the cow dung or horse dung, any dung, that is cow dung. Okay, that fungi is called Corpophilus fungi. Pilobolus. You can notice this fungi also. Our cows are putting the cow dungs anywhere, and two days after you can see on the upper layer somewhat powdery like substance in black color. That is called Corpophilus fungi. It is a saprophytic fungi, decaying the cow dung and making it as an nutritious to the soil. Enrich the soil nutrients with the help of decaying the fungi. Okay. Mycelium is branched and cenocytic. So the mycelium here, it is branched, and cenocytic means the branches you can see, and the cenocytic means there is no cross walls in the mycelium. When you took the cross section, you can see without cross walls the nucleus is dispersed, scattered. Asexual reproduction. So as we study about the asexual reproduction, it takes place by spores, and here the spores are produced in the sporangia. And sexual reproduction by the fusion of gametangia, and it produces zygospore. The first zygospore is formed in this class, hence the name came as Zygomycetes. So this is in this class only the first zygospore appear. The fusion of two gametes forms the zygote. So because of that only Zygomycetes, the name arises. So zygospore, after the zygospore formation, the zygospore undergo meiosis and produces spores in the sporangia. From the sporangia, the spores are zygosporangia and the spores are liberated. So this is a picture of rhizopus, that is the rhizopus. So the stalk, this is the sporangium, and the stalk is called sporangiophore. So stalk of the sporangia is called sporangiophore. And this portion, this portion, this is called columella. And this is the bag-like structure is called sporangium, where the spores is kept inside until the maturity. After the maturity, the spores will be liberated like this. Okay, so this is the characteristics of Zygomycetes. Next we move on to Ascomycids. So some of the examples of Ascomycids are yeast, powdery mildews, cup fungi, morels, and so on. You're going to see. And this is a Pezisa. It is a cup fungi. And other three, this one, and this one are the fruit body. Clystidium is a closed fruit body. and that is perithecium. Perithecium, you can see a flask shaped. When you go to the chemistry lab, you can see some of the flask shaped glasswares you can see, that is called like that it is having, no. So it is perithecium, flask shape, but it has an opening. But clystecium is completely closed and apothecium is cup shaped. Cup shaped. These are the fruit bodies we will discuss discuss after. First, the habitat of the fungi. Mostly they are terrestrial environment. Some are aquatic. So mostly they live on the land, and some are aquatic. The aquatic may be freshwater or marine water. And the mycelium is well developed. Branch with simple septum. So the septum means there is cross wall. In Zygomycetes and Oomycetes there is no cross walls. So they are cenocytic. But here the cross wall is present. So it is septate. And majority or saprophytes, that is the mode of nutrition is saprophyte, means they live on dead organic matter and they take the food as the dead organic matter. And the parasites. So parasites, they live in another organism and creates disease to the organism. That is example is powdery mildew in Erysifa. Asexual reproduction takes place by fission, budding, oidea, gonorrhea, chlamydospores. So by all the spores, the asexual reproduction takes place. And sexual reproduction, here plasmogamy. As we have discussed this plasmogamy in methods of reproduction. Plasmogamy means what? Fusion of protoplast. After the fusion of protoplast, what has to be taken place? Karyogamy has to taken place. That is, fusion of nucleus has to take place. But there is no karyogamy. So fusion of plasmogamy takes place, but nucleus won't fused. So due to that, the hyphal condition is dikaryotic. So two nucleus in a cell. It looks like a two nucleus in a cell. A special hyphae called Ascogenous hyphae is formed. Due to this, a special hyphae is formed, that is called Ascogenous hyphae. So a crosshair is formed when the tip of the Ascogenous hyphae is curved, forming a hook-like structure. You can see this is the Ascogenous hyphae. The tip is slightly curved. Okay, this is called crosshair hyphae. The two nuclei in the penultimate cell of the hyphae fused to form diploid nucleus. So it forms. So while crosshair hyphae, here the cross walls are destroyed and it looks like a two nuclei. It looks like a two nuclei, that is like diploid nuclei. This cell forms the young ascus. So like that, when it folding, it forms the two nuclei become four nuclei become eight nuclei, like that. It forms the ascus like structure. Okay, and it is called the young ascus. The diploid nuclei now undergo mitotic, that is meiotic division. So this undergo meiotic division and forms the four daughter cells. This four daughter cells results in 8 ascospores. So they further divide and form, that is till the 4 it is meiotic. After 4 that is meiosis over, from this stage to this stage mitosis takes place. From the mitosis you are getting 8 ascospores. 8 ascospores and nucleus get organized into 8 ascospores. Okay. The ascospores forms a bag like structure. So this is this forms a bag like structure. So this is called ascus. Ascus. So the ascus is seen. It is in a bag. So okay, bag. It is. So it is called as, this fungi is called as sac fungi because the ascospores are inside a bag like structure. That bag is called sac. It looks like a sac. So it is called as sac fungi. Acai is surrounded by a sterile fungi, sterile hyphae, forming the fruit body called ascospores. So as we discussed in the first slide, that is here. So the ascus is surrounded by a sterile hyphae and it forms the fruit body. And based on that, it is divided into three types. Clystium. Clystium means, clystos means closed. So here there is no gap. Completely it is closed. So closed and round in shape. And perithecium it is flask shaped. Apothecium it is cup shaped. The fruit body is only here it is completely closed, and in flask shape you have the opening here, and cup shape the entire thing is exposed. In between that the sterile hyphae and the fertile hyphae with ascospores are seen in the cup shaped or flask shaped or closed type of fruit bodies, that is clystium, perithecium, apothecium. Next we move on to the Basidiomycetes. Puffballs, toadstools, bird's nest fungi, bracket fungi, stint horns, rust and smut. These are the Basidiomycetes fungi. Okay, the example you can see here. Dolichopore septums. It looks like a dumbbells, no? Each dumbbell come and forms a dolichopore septum, which is used in the fertilization, sexual reproduction. Okay. And it is a clamp connection. Clamp connection we will discuss when we discuss about the sexual reproduction. So here also the mycelium is well developed, and it is septate with dolichopore septum. So this is the dolichopore septum, and it has cross walls. Okay, you have cross walls here and all. You have cross walls. Okay, so this is the dolichopore septum. So there are three types of mycelium: primary, secondary and tertiary. Primary means only one per septum, one nucleus will be there. So in alpha there is a septum, because it is a septate. So one nucleus is seen from each cell, like that. Secondary means dikaryotic, 2 nucleus. Tertiary means 3 nucleus. Okay, like this. And clamp connections are formed to maintain dikaryotic condition. So this is the clamped condition. So the clamp is an outgrowth was developing. Due to that, the 2 nucleus will go here, or this 2 nucleus come this sides, like that, to maintain the here 2 nucleus are. So here 2 nucleus and one will come. So this 2 come one side and this 2 other side. To maintain the 2 nucleus condition, the clamp connection is used and it is formed. Asexual reproduction takes place by gonidia, oidia or budding, and sexual reproduction is present but sex organs are absent, that is androecium and oogonium is absent. Stomatogamy or spermatization results in plasmogamy. So stomatogamy and spermatization will take place, but here it results in plasmogamy. Due to the plasmogamy, the fusion of protoplast takes place. Due to the plasmogamy, the dikaryo phase is retained, because the karyogamy delays here also, as Ascomycids. Here also the karyogamy won't takes place. That is, it takes place very late. Due to that, the phase possesses prolonged dikaryo, that is 2 nucleus phase. And karyogamy takes place in Basidium. So Basidium is a kind of spore formed from the basidiocarp. So Basidium, so because of this Basidium spore only, the class name is Basidiomycetes. So karyogamy takes place in Basidium and it is immediately followed by Meiotic Division. So it is immediately after Basidium, that is karyogamy takes place and in Basidium, and after that the Meiotic Division takes place in a sudden. The 4 nuclei thus formed are transformed into basidiospore. So the 4 nuclei are formed. They are formed to basidiospores. Okay, and it forms and where it is formed? It is formed on the stalk called sterigmata. So the sterigmata is outside. So it is exogenous in origin. The basidium is club-shaped with 4 basidiospores. And the group, it is called as club fungi also. The Basidiomycete fungi is otherwise known as club fungi because they form a group-like structure with 4 basidiospores. That shape looks like a club, so it is called as club fungi. The fruit body formed first is the basidiocarp. Here the fruit body is basidiocarp. Carp is the fruit and basidio is the spores. So it is called as basidiocarp. Next we move on to the Deuteromycetes or fungi imperfect. So, Pycnidium, Ascomycetes, Synema. So the fungi, this fungi lacks asexual reproduction. So there is no sexual reproduction in this fungi. So it is called as imperfect fungi. So most of the fungi are saprophytes. So most of them are saprophytes, live on dead organic matter. And some are plants and animals are as parasites. In some plants and animal parasites, that is this fungi is living as a parasite in plants and animals. Asexual reproduction takes place by gonidia, chlamydospores, budding, oidea, etc. So the gonidia has a special structure called, here they will call it as pycnidium, acervellus, sporoducium and simneta. So this is pycnidium, acervellus and simnema. So like that special kinds of spores are also produced by gonidia. And parasexual cycle. Parasexual cycle means it looks like a sexual phase, but there is no formation of sex organs or sexual reproduction doesn't take place. And there is no meiosis also. The meiosis or gamete formation is not there in this, but it looks like a sexual phase. So that kind of sexual phase is called as parasexual phase. And due to this, it brings the genetic variation in some of the species of Deuteromycetes. Hope you understand the classification now. So how many subdivisions and divisions are there in classification? If they ask which was classified by Alexopoulus and Mims, means 3 main divisions and 8 subdivisions and 11 classes. Traditional classification, it is 4 classes. So hope you understand well the classification. If you have any doubts, post your doubts in the comment box. Thank you for watching the video. Give a thumbs up, share and subscribe to Science Easy Tech channel.